Welcome everyone to this morning's webinar, TEL-Seq Data Visualization using IGV. My name is David Nordell. I'll be your host today, and with that, I'll go ahead and hand it over to our presenter this morning. You?
Yeah, thank you. And today I'm going to talk about TEL-Seq data visualization using IGV. And before we start, I would like to clarify the definition for some names. So what is the TEL-Seq? What is the TEL-Sort? And what is the IGV? TEL-Seq is the transparency-based library preparation kit developed by UST that enable Illumina sequencing platform to produce a super long-range genomic or transcriptomic data. And what is the TEL-Sort? TEL-Sort is the software developed by UST to process and analyze the TEL-Seq sequencing read so that the user may retrieve the facing information from the linked read. And what is the IGV? The full name of the IGV is the Integrative Genome Viewer. It's an open source integrated tools developed by the Broad Institute that enabled visual exploration of the genomic data. And we use the IGV to show our TEL-Sort result. And in this presentation, we have a four part. The first part is the requirement. The second part, I'm going to describe how to convert the TEL-Sort result to the IGV tracks. And the third part, I'm going to describe how to upload to the IGV and then interpret the IGV track. And the fourth part is an example of the TEL-Seq data visualization. And this is amplicon-based TEL-Seq. And this slide showed the scope of this presentation. As I just mentioned, I'm going to cover how to convert the TEL-Sort output files into IGV tracks and also upload them to the IGV browser and interpret them. However, I'm not going to present some general stuff. For example, how to use a Linux command line to run the scripts on the server. And also, I'm not going to cover how to run the TEL-Sort to generate the TEL-Sort output files. This is the upstream of the current analysis. And also, I assume the user already download and install the IGV in their local computer. So before we start, we need some document related to this analysis. So please go to our website and then use a resource tab and click the TEL-Seq software tab. Then this HTML page will be on your browser. Please click this big button to download the software related to this analysis. And after you download, you will notice this is a zip file containing the three elements. The first element is the PDF file. It's the user guide. And actually, this is the writing version of this presentation. And then the second item is the Perl script, which we are going to use this Perl script to generate the IGV files. And then the third item is the examples of the IGV files generated using this Perl script. Now, we already have that Perl script. We need to run that Perl script. However, that Perl script needs some dependencies. They are WhatsApp, SAM tools, ECF tools, and HTTPS lib. So if your server has not installed those dependencies, please follow those links to download and install them on your server. And also, please update your path variable so that the Perl script will automatically locate those dependencies. And also, do not forget to install IGV on your local computer. And if you haven't done it, you may just follow this link, download and install it. And now we have Perl script ready. We have dependencies ready. Now we are ready to run that Perl script. However, before that, we need to know which input file. That means what is the parameters for those for the Perl script. So as I mentioned, so this Perl script will convert the tail sort result to the IGV tracks. So that means the tail sort result will be the input. So if you provide the path to the directory that contains the tail sort output, and also provide the prefix that has been used for the tail sort analysis, then when you provide these two parameters, that Perl script that will know where to locate those BAM files, those VCF generated from the tail sort. Also, the Perl script required the reference genome for the argument. So this reference genome is also being used for the tail sort data analysis. So basically, you provide these three arguments, then that Perl script will generate the IGV files. And here is the syntax. This Perl script requires this tail sort data directory, prefix, and reference genome it uses. Then that's Perl script will generate the IGV files for you. There's the fourth argument, which is the maximum cores. How many CPUs you would like to use. By default, if the user doesn't provide the CPUs, then the 30 CPUs will be automatically being used. For this Perl script performance, this is depend on the data size and how many cores you used. For example, if we have about like 1 billion pair-end read from human, and then we configure 30 cores, the process takes about 2.5 hours for the running. Now, I assume we already successfully run that Perl script. If so, under the tail sort directory you provided to the Perl script, you will have a subdirectory with the suffix of _IGV files as the subdirectory. For example, if you provide a prefix 501, then under the tail sort data directory, you will generate 501_IGV_ files subdirectory. And under this subdirectory, all the results from that Perl script will save there. Now, you already generated the IGV files. What you need to do is upload to the IGV. So first, you need to transfer that data from the server to your local computer, because this Perl script is located on the server. And after you run, that the result will save in the server. You need FTP back to your local computer. Assume you are in the FTP back. Now you start IGV browser. And then the first thing you need to do is select the appropriate reference genome. For example, in our example, it's a human genome HG38. So you also need to configure in your IGV browser to use the HG38. And after that, you upload your IGV tracks. And you go to here, File, and then you select the load file from. Then there's a pop-out window will be generated. And then you navigate to the data files you just FTP back to your local computer, and then load them one by one to your IGV browser. So total, there's 4 types of files there. GTF file, which is represent the face block, the face BAM file, which is read aligned to the reference genome, and the read being phased. And then the VCF file, which is the variant and the genotype, and then the pseudo BAM. So we will going to talk about the one by one. So after you upload those tracks, please tell the IGV which genomic location you would like to view. For example, you are interested on the HLA-C gene, then you just type HLA-C, click go or type enter. Then the IGV browser will bring you this display. So all those 4 tracks will be loaded here, 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 and here. And before we go to each track one by one, so I would like to present a tip for the IGV. You see this icon. If you click this icon, then the IGV will give you an option how are you going to show the detail. For example, in our case, I select to show the detail on click. That means if I click this item, for example, this one, then I will get the information. There's a pop-out window showed here. Now let's go to the each track one by one. The first track is the VCF track. The VCF track is located here. And if we enlarge, we will show something like that. So there's 2 subtracks. Here is the blue and the red color vertical bars that represent the variant come from the samples we analyze. And there's another only short blue bars. This is the genotype. So both tracks come from the VCF file. And if you want to the details, just click and then you will generate this pop-out window. This one is for the variant. This one is for the genotype, variant genotype. Okay, then the second track called this pseudo-BAM track. The pseudo-BAM track is the most important track for the TEL-seq result. So it displays the face, the variant, as the vertical bars connected by the seq line and the numbers, just like this. So this is a variant connected by the seq lines, and in the middle there's a number. So the thick lines connect this phased variant together, so in the same haplotype. Then the number represent the distance between two adjacent variants. For example, in this case, there's a 1018 nucleotide between this variant and this variant. If we enlarge this portion to this one, if we enlarge this portion, this portion, this portion, and this portion, we will show something like this. So this means this C, this C, this C, and this G is linked together, is represent one haplotype. And this gray box, this gray box, and this gray box, and this gray box connect together. This form another haplotype. And you wonder why is just a gray box, not the nucleotide. So this is because the IGV by default, if the nucleotide here is agreed with the reference genome, then that nucleotide character will not be show up. In order to show up that character, you may just right-click your mouse on this track, and then choose show all the bases, and those bases will show up. So in this case, so this 4 gray box will be the TTAC. So those TTAC is linked together. And this is the phased BAM track. The phased BAM track contains the aligned read with the phasing information. The phasing BAM contains the two customer tags. One is HP tag and another is the PS tag. HP tag assigned with the values of 1 or 2 represent haplotype 1 or haplotype 2. The PS tag assigned the value with the phased block ID, which is the core algorithm. This ID is assigned by our core algorithm HyperCut2. And this in this track is a multiple view option IGV allow. There's a very informative view option. So the people may would like to have all the read belong to haplotype 1 grouped together, aligned to the reference genome, and the same as haplotype 2, and also the unphased read. So to do so, you may just right-click this track, then select the linked read view MI, which means the molecular index, and also select the squish. So after you select these two options, then the layout will display something like that. And this is the last track, it's called the GTF track. So the GTF track is for the phased block. So why we use this GTF track? Let's assume if you have two phased block adjacent, however, there's a broken in the middle or is not being phased, then you may have an issue. The read in the haplotype 1, the read in the haplotype 1, whether they belong to the same haplotype, maybe not, just because there's a broken. So the read here belong to the haplotype 1. It's only valid in this phase block. Same thing like this, same thing in this phase block. So if your read assigned to the haplotype 1, this is only valid in this phase block. So in order to let the user to not get confused, so we need phase block so that the user will know if they are focusing on this region, they are safe. They don't generate a switch error. But if they don't show this part, they may have generate a switch error because the read belong to the read show the haplotype 1 may not belong to the same haplotype. Okay, now let's go to an example using the IGV. So we have a PI3 kinase in the cDNA level, and then we PCR amplified that region, then we generate the amplicon. So that region contained 3 SNVs, located here, and then located here. And the phasing result is strongly linked to the breast cancer. So we would like to have that phasing result. Then we generate that data, showed something like this. Because this is the cDNA level, we would like to use that PI3 kinase amplicon as the reference. And then we upload our pseudo-BAM track, we upload our BAM track, we upload our phase block track. And if we enlarge this portion, you will see something like this. So this G linked to this A, linked to this reference, this reference linked to this C. And then this is show the detail of in the read BAM track. So you also can have the multiple samples displayed together. For example, here shows the 2 samples, and here is 1 sample. Here is another sample. And we have a pseudo-BAM track, read track, haplotype 1, haplotype 2, haplotype 1, haplotype 2, and then phase the block. So what is the conclusion here? So we can easily to view the tail sort result use the IGV, but in the current status, this piece of information is separated from the tail sort, and in the future, we would like to embed this process in the tail sort pipeline. Then later, the user will not need this step anymore. The automatic generated from the tail sort. And if you would like to learn our software, learn more information from our software, please visit our website, then follow the link. Or if you want to get more detailed information about IGV, please visit the Broad Institute website. And also, if you are interested to our application node, please visit our website. And we have a PI3 kinase, and we have a sodium channel have a sodium channel related TLC data. And also we do the facing with the black 1, black 2. And if you have any questions, please contact our technical support in our company. Thank you.
Thank you. And with that, we'll go ahead and open it up to any questions. If you do have any questions, please go ahead and type them in chat. As a reminder, this webinar is being recorded and a copy of it will be emailed out. So if you or anyone that you would like to share it with missed it or missed any part of it, you will be able to rewatch it then. And I will go ahead and enable mics for the attendees as well, in case you prefer to ask your question verbally. So that should be done. So again, if you have any questions, please go ahead and just enable your mic and ask them verbally or type them in chat. And we'll give it just a couple minutes. All right, well, it doesn't look like we have too many questions coming in right now. I'm sure there will be some that people have once they start getting their hands on it and using it. Again, if you do have any questions, please email technicalsupport@universalsequencing.com. If you'd like any help on orders or what you need for actually, getting your hands on the library prep kit, please email sales@universalsequencing.com. With that, we'll go ahead and wrap it up. Once again, thank you all for being here, and I hope you have a wonderful day.
Thank you.